I'm Nick. I work in Malkova Lab, and I'm presenting on DNA repair. So, DNA gets damaged a lot, and it can happen from many things such as X-ray, getting an X-ray, sunlight, different drugs, things like that. And one particular type of damage is what's called a double-stranded break. And a double-stranded break is when both strands of the chromosome are essentially cut in half. And over time, cells have developed different pathways which repair this break. And they're quote-unquote good and detrimental pathways. And an example of a good pathway would be what's called gene conversion. And basically what gene conversion does is that it just copies just the part, the broken part from a homolog chromosome, so it copies what it's missing and it'll replace it into the broken one. And then a different pathway, quote-unquote, a bad or a not good one, is what's called break-induced replication. And break-induced replication works by invading a homolog chromosome, and it doesn't just copy the part that's missing, it copies it all the way to the end. So this can be almost the entire length of the chromosome, and this leads to high mutation rates. And so this brings me to my first question, which is how does a cell choose which pathway to use? And what are the underlying mechanisms behind that, and like how it channels it into one versus another? And this actually indirectly leads me to my second question as well, is that what is happening in G1 and S phase of the cell cycle? Because break-induced replication has been studied extensively in G2 of the cell cycle. But I aim to study it in G1 and S phase, and I plan to do that because that is where most cells, or non-dividing cells, spend most of their time. And in order to study it in G1 S phase, I plan to mutate a yeast cell to hold it in G1 S phase, and then to use what's called a inverted repeat, which is basically just essentially a loop on the end of the chromosome from the break site, so it'd be right here. And basically by doing that, I hope to channel the break into BIR and better understand the mechanisms behind that. Because in G2 of the phase, it's known that BIR is very bad. But since if it's happening in G1 and S phase, it might be different, and it might actually be better than what's expected. So by doing that, I hope to better understand the mechanisms, and if it is different, what the differences are and why it is different in each cell phase. So, thank you.